we do have a paper that is out. It was published in 2002. It was a collaboration with scientists and stakeholders who had actually been working on this for a number of years. And it came together as the first summary of ocean acidification in the Southeast region. In that paper, we determined a list of priority monitoring sites, with the top 3 being Sapelo Island, Georgia, Gray's Reef National Marine Sanctuary, and the Biscayne National Park at the northern edge of the Florida Reef Tract. We are working on getting a monitoring site down in the Florida Keys that will hopefully come within the next year, that can partner with a NOAA site that's already down there monitoring. I'd like to also introduce you to the executive committee that is part of SOCAN, and that includes Deborah Hernandez, who is the executive director of SECOORA. We also have Laura Corman, who you've just met, who's new this year with us as a program coordinator for SECOORA. Myself, where I'm actually based at Mote Marine Laboratory in Sarasota, Florida, and Dr. Janet Raymer, who is based at the University of Delaware, but does a lot of coastal and ocean acidification work, especially at the LTER site, the long-term ecological research site in the Georgia Coastal Ecosystems region. So the purpose of this meeting, one of the main purposes of this meeting, is to provide information to an interagency working group on ocean acidification. And this is made up of partners from the NOAA Ocean Acidification Program, as well as other groups. They have actually reached out to all of the CANs. There's a number of CAN networks. So SOCAN is just the Southeast. There's also GCAN in the Gulf of Mexico, and others throughout the rest of the US. They've asked us to contribute to a vulnerability report. And this is a report that is going to be presented to Congress to show where societal vulnerabilities to ocean acidification exist or are emerging, in order to provide actionable information for marine resource decision-making and to bolster the resilience of the nation's blue economy. And so we are hoping that this workshop will help provide information on these very specific topics. We were given an outline by this group, and so these topics include exposure, biological response, and social vulnerability. And those will be the 3 main themes that you'll see throughout the meeting today. And then so we will hopefully be writing up a report after this workshop with everybody's input who is participating. So please free to be, especially in the afternoon sessions, we're looking for some good, strong participation to help us put that together. So with that, we'll go on to the next slide. I'm going to go ahead and introduce our first speaker. Janet is a co-coordinator for SOCAN and an ocean acidification researcher at the University of Delaware. Next slide. She's going to be talking about the general state of what's going on in the Southeast region. So Janet, take it away.
Thanks, Emily. And I'm really happy to be here this morning, giving you a general background of acidification and the Southeast region as it relates to acidification. Next slide. So in our region, we have a wealth of diversity, and most of our region seems to be covered with grass beds and salt marshes, which you can see in green and orange. We also have shallow corals and hard structure reefs, as well as deep corals and soft bottom reefs. So we have this, sorry, I forgot mangroves. So mangroves in southern Florida, I won't actually be going into today because we don't have as much research on mangroves as we do with some of our other ecosystems, but we're getting there. We want to really understand how resilient our ecosystems and societies that depend on ecosystems are. Next slide, please. So acidification in coastal waters has been defined as a decrease in pH and carbonate mineral or carbonate ion saturation because of an increase in dissolved CO2. This CO2 can come from any number of different sources, including CO2 from organic matter breakdown by bacteria or a process we call respiration, CO2 transported from rivers, estuaries, and other coastal waters to the shelf, CO2 dissolving into the surface from the atmosphere, and with burning of fossil fuels, that's usually a pretty big impact, Also, currents can bring high CO2 water either from upwelling off of the shelf. In our region, we have a very wide shelf, so that might not be as influential in the coastal waters, but any kind of alongshore currents can also transport CO2 from one region to another. Finally, thermodynamic changes such as temperature increase can also cause the partial pressure of CO2 to increase. We're not going into gas laws today, so no need to panic. Next slide, please. The Southeast has extensive watersheds, and I included in this map the Gulf of Mexico region as well because of the loop current that connects us to our partners in to the west of us with GCAN. We do work closely with them, and we hope to work even closer with them in the future. As I mentioned, fresh water is brought to the coastal zone, so it's not just where it's raining at the coast, but also it's very important what's happening in the upper portions of watersheds. Extensive watersheds in the southeast have an important impact because freshwater has lower pH than coastal waters, and freshwater also tends to have high amounts of organic matter, and it is this organic matter that can be respired either as it's traveling through the watershed or once it reaches the coast to release even more CO2. The Intergovernmental Panel on Climate Change has predicted wetter climate conditions in the southeast and more frequent and stronger tropical cyclones through the next century. So one of the things we want to understand is how freshwater entering the coastal zone and how climate changes can impact coastal acidification and the resiliency of communities. The saving grace could be, I underlined could there, that the warm, salty subtropical waters of the Atlantic have a high buffering capacity. This means that these warm, salty waters could help buffer or protect us against acidification. Next slide, please. Without getting too in depth into the chemistry of acidification, I just want to point out that there are two main reactions that determine the pH and carbonate saturation state. In this equation, the first thing that we have to assume is that CO2 is absorbing into the surface from the atmosphere, as well as being produced inside the water. So carbon dioxide is going to react with water and any free carbonate ions. This forms two bicarbonate ions, and this is what essentially impedes the calcification process. So without carbonate ions, corals and other shellfish cannot form their hard structures. So when the carbonate ion is consumed or is not available to animals, we call this undersaturation. Next slide, please. In the second reaction, excess CO2 in an undersaturated state with carbonate in an undersaturated state reacts directly with water. This forms hydrogen protons that are free floating and also forms one molecule of carbonic acid. Now, you might think, well, if the two equations are occurring simultaneously, does this mean that two reactions are impeding the process? Yes. So first, carbonate mineral formation is impeded, but then you also have acidification of the water due directly to the hydrogen protons. So when we talk about acidification, we talk about these two reactions concurrently. Next slide, please. So now that I've given you a little bit of background on the region in general and some of the chemistry, I'm going to give you some examples of some recent research and a little bit of some insights into what we want to accomplish today with this meeting. Next slide. So I'm going to start down in the Caribbean and the upper Florida reef tract, where temperature increases and biological release of carbon dioxide are driving the pH decrease and the carbonate undersaturation. These studies were conducted off of the southwest corner of Puerto Rico and up in the upper Florida reef tract, which is that yellow star on the map. So what's happening to corals? Well, we know what's happening in the wild, but does that actually translate to laboratory studies and vice versa? In laboratory studies, it's been found that some corals can have decreased growth when they're grown in controlled conditions at a pH of 7.7 compared with typical ocean pH, which could be around 8.2. Courtney Claypock is going to give us a little more information on this later this morning, but I just want to point out that this photo is provided by Emily Hall at Moat, where they're conducting a lot of mesocosm studies. Next slide, please. Another research track that has been gone down in the Florida reef track is the study of elevation changes. So you might think, well, okay, corals may not be forming as much, but what does that really have to do with our coastline? Well, in the upper section of the Florida reef track, we've seen dissolution, or what we call negative net community calcification, since the 1930s. This essentially means that reefs are dissolving in simple terms. This elevation change has been up to almost 11.5 meters, which means we have 11.5 meters less of reef in some places, and it seems to be a little bit stronger in the northern section. This can be caused by seasonally negative low pH and carbonate undersaturation, but these seasonal changes naturally occur. However, if you have climate change on top of these, you can have elongated periods of negative reef growth, which could influence longer term trends. Loss of reef structure can actually directly impact the amount of energy in the waves that hit our coastline. So reefs are not just pretty things to look at. They're not just a cultural ecosystem service, but they actually have value, economic value, when it comes to human structures. So the USGS, NOAA, and other government agencies have been looking into the value of reefs as it pertains to protecting our coastlines. Next slide, please. Pamlico Sound in our northern region is actually a very restricted body of water with limited exchange with shelf waters. Extreme weather events can enhance CO2 and organic matter in the sound and cause dramatic pH decreases due to freshwater influx, but also organic matter breakdown. You can see in this graphic that pH was dramatically lower after the passing of Hurricane Florence back in 2018. pH in the upstream portion, which you can see in blue on the September 26th graphic, is due to that high influx of fresh water, but also any kind of disturbance can churn organic matter up into the water column from the sediments, which can then undergo bacterial respiration, thus releasing CO2 into the water column. So this is likely due to two factors, one being the lower pH of freshwater influx into the sound, but also this increase in organic matter, thus the decrease in pH. Remember that the IPCC has predicted an increase in tropical cyclone activity throughout the Southeast in the coming century. So it's unclear right now how these low pH conditions can affect Pamlico Sound and other enclosed bodies of water. And also remember that the waters on the South Atlantic Bight Shelf could have a high buffering capacity against acidification, but we have yet to truly understand how the shelf waters and coastal waters are interacting. Next slide, please. So now I'm going to take us down to a trip to Tampa Bay. Tampa Bay is an urban estuary. Urban estuaries are threatened by land use change. As residential and industrial usage increases, the amount of excess nutrients into urban estuaries increases. These nutrients can also cause increased plant growth and algae production, therefore more organic matter production and more organic matter breakdown, releasing CO2. So you have two processes here, CO2 uptake, which we're happy about, but then excess CO2 release into the water from the excess organic matter that's being produced. So we want that to balance out. We don't want one to be more than the other to maintain a happy ecosystem. In the 1980s, unfortunately, this very issue existed in Tampa Bay, where you can see in this picture an absolute overgrowth of algae and other plants. However, in recent years, the mitigation of excess nutrients entering the bay and restoration of natural seagrass beds has helped increase the pH, counter acidification, because seagrass communities absorb high amounts of CO2. We also see this in the Chesapeake Bay, in our the region to the north of us, our partners with the Mid-Atlantic Coastal Acidification Network. I hope some of you have joined us today, are studying seagrass beds as well throughout the East Coast. Ed Sherwood is actually going to give us some more information on Tampa Bay later on this morning, so we'll wait to hear some more about that from him. If you could go to the next slide, Laura. Now, my own passion project, obviously, is the South Atlantic Bight. In South Atlantic Bight coastal waters, dissolved CO2 could be increasing at a rate of up to 4.5 microatmospheres per year. To put that in perspective, it's about double the atmospheric increase, which is 2 microatmospheres per year, give or take a little bit. This increase is due to CO2 from atmospheric inputs across the shelf, CO2 from the breakdown of organic matter, and low buffering capacity of fresh water onto the shelf. In this graphic, I actually analyzed 5 different locations. The outer South Atlantic Bight shelf, which is considered to be 200-meter depth and outward. The middle Atlantic Bight shelf, which is 60 meters to 200 meters. The inner shelf, which is about 15 to 60 meters depth, which is also where the Gray's Reef mooring is located, which is a high frequency CO2 time series, which we'll talk a little bit more about. And then the intercoastal waters, which would be anything from 15 meters and shallower. So while the CO2 increase is not exactly the same, it's all trending in the same direction across the South Atlantic Bight. The resulting pH decrease could be as much as 0.004 pH units per year. Remember, the pH is on a log scale, so this is quite a significant increase compared to open ocean estimates, which are 0.002 units per year. Given the high buffering capacity of subtropical Atlantic waters, we don't quite know yet what the impact of this decline in pH is on coastal ecosystems, communities, or industries. And that's one of the things that we will be focusing on in the vulnerability report. Next slide, please. So the first step in answering questions about vulnerability and resiliency is to understand the state of acidification. We have monitoring sites across the Southeast for CO2 and pH changes. All of these sites, we have collaborators, and I should say SOCAN is not the owner of these sites, but we try to work closely with each and every collaborator so that we can synthesize data across the Southeast. So right now we have moored CO2 and pH time series that measure every 3 hours at Gray's Reef, which is off the coast of Georgia. There's the Tampa Bay time series. There's another time series at Chicha Rocks, which is in the Florida Reef Tract. And I also want to take this opportunity to mention that there is another time series in coastal Louisiana waters that actually falls within the GCAN region. But given the loop current in the Gulf of Mexico, it's very important to understand transport processes. So we will be working with GCAN even more closely in the future to try to understand this horizontal transport. There are also pH sensors at 6 national estuarine research reserves and 3 national estuary program sites throughout the Southeast. There's also the discrete sampling time series from Georgia Coastal Ecosystems, which is around Sapelo Island and in the Georgia marshes. And there are 20 sites for CO2 and or pH from the MODMON project in the Neuse River Estuary in Pamlico Sound. Much of this data is available from SECOORA.org, but also through the National Centers for Environmental Information. In 2017, SOCAN hosted a workshop to establish what our priority sites would be, and those are the numbered sites in the map. I will point out that while the green circles, which were not identified in 2017 as the high priority sites, have actually been selected for funding. And once the funding becomes available in the coming fiscal years, we hope 2022 and 2023, so SOCAN will be directly involved in research projects at those sites. Next slide, please. So the big question about vulnerability is, what do we know that we don't know? Because we have a lot of data on species specific and mesocosm studies and what's going on in some ecosystems, but we really don't know yet how that affects society. And when we talk about society, we're looking at things from the industrial perspective, from cultural groups, and our overall general well-being. So what is general well-being? Well, you have the human dimension, which includes adaptive strategies, socioeconomic impacts, and education and outreach. So that's when I mentioned dissolution of coral reefs and the impact on our coastlines. Well, what is that economic value? How much do the reefs protect us? Or if we're talking about industry, how does it affect shellfishing industry and the aquaculture industry? We also know that acidification does not exist in a bubble, and that there are multiple stressors that can enhance or potentially counteract acidification. Some of those stressors, which we're calling multi-stressors, are sea level rise, temperature rise, harmful algal blooms, and hypoxia. Finally, we want to understand how extreme events such as storms, droughts, and floods impact coastal acidification. I mentioned that as the water column is stirred up, there could be churning of organic matter from the sediments into the water. Well, how is that going to be increased or mitigated through climate change and human mitigation methods? Or how is a drought going to affect the fresh water entering the coastal zone? Does that mean that acidification may not be as pronounced one year compared to another due to extreme precipitation or drought? There are a lot of questions that we still haven't answered, but we're certainly on the right track to be able to fund new projects in the upcoming years to help understand acidification in the Southeast even better. And that's all I have.
Thank you, Janet. Hi, I'm Kim Yates, and I'm going to be moderating questions for Janet's presentation today. I'm with the U.S. Geological Survey and also a member of the Interagency Working Group on Ocean Acidification. If you have any questions for Janet, please place them in the chat. And Janet, while we're waiting for people to enter their questions or think of questions, I have a question to start out for you. What's the biggest challenge to monitoring and understanding acidification chemistry and dynamics in our region? Seems like a lot of challenges.
Well, I would say the first challenge would definitely be the heterogeneity of the coastal zone. So I mentioned that our region has a number of different ecosystems, and so we can't study one place and accurately upscale or translate that finding to multiple places within our region. There are currents that affect pH and carbonate saturation around the Southeast. There are temperature changes from one region to the other, salinity changes, which has a direct impact on buffering capacity. Freshwater is not homogeneous throughout our region. So just being able to get a good understanding of the different ecosystems, I think, is the first challenge. I think the really obvious answer to the question is also funding, because without funding, we can't establish long-term sites. Long-term sites are needed because you have this natural juxtaposition of seasonal cycles, but also long-term changes. And you have to be able to separate the two. And so that also takes manpower, which is one of the most expensive aspects of acidification research, and just really having good partnerships, which SOCAN is working towards throughout the region.
Thank you. Does anyone else have any questions for Janet? If you'd prefer to unmute and ask a question, that's okay too, or otherwise put it in the chat. Let's see, I have a couple of questions that have popped up. First one is, can you give some examples where groups can find funding for coastal acidification work?
Sure. So there's the Ocean Acidification Program through NOAA. Recently, I think there were 2 calls. Usually calls go out I want to say the end of the summer, beginning of the fall. There's the but the state sea grants. NSF has usually bigger projects through NSF, but recently SOCAN has received funding through the IUSE program, which is directly funds SECOORA, and we also get money from OAP, and we've also had some good luck recently with state sea grants.
Okay, and another question from Jinping Hu. For the reef accretion and erosion along Southeast Florida, is there more roughness along the reef track so the water exchange between inside and outside the reef track has become more frequent?
That's a really good question. And I have to say the best person to answer that would be Kim.
So thanks, Janet.
Yeah.
What we're finding is that as reefs degrade, we actually lose roughness, or what we call topographic complexity. And that does allow for more exchange. And more importantly, what we find is greater energy dissipation of waves at the shoreline, which causes coastal erosion. So that's sort of the short answer on that one. And another question, is there any potential for citizen science to help, or is there a technological barrier to that?
That's a really great question. The Georgia Coastal Ecosystems Project actually does have a gentleman who is our citizen scientist who collects water in the upper portion of the Altamaha River above the head of the tide, so that we can get that freshwater end member to understand what is coming into the coastal zone through the watershed. The drawback of citizen science in acidification research is that we have to preserve or poison water samples with mercuric chloride. And so there can be some red tape with getting that chemical and having that chemical be used by a citizen scientist. However, I do know that there are other citizen scientist programs at Coastal Carolina University that we hope we can bring into acidification research. We're going to need a little bit more time to figure that out. So yes and no.
Okay, and last question. And before I ask this last question, if you have any other questions, please write them down and we can address them in the breakout sessions this afternoon. So last question is what are the biggest needs / priorities specifically for understanding the role of mangroves so they can be included next time you give an excellent presentation such as this?
Thanks, Ryan. I would say monitoring sites. We need to have instrumentation purchased and set up, and someone to maintain the instruments. It kind of brings to mind, we need more collaborations, and we need to have that ability to set up, you know, whatever the best instrumentation would be for mangroves. There have been several studies on CO2 exchange or fluxes in mangrove, but to my knowledge, we don't have any definitive long-term or seasonal acidification studies. So I guess the biggest priority is to reach out and try to find collaborators.
Okay, thank you, Janet. And I will turn it back over to continue with the next presentation.
Thanks, Kim. Janet, thank you. That was amazing. That was really good. And that does conclude our session on exposure, current and future levels of ocean acidification. So now we're going to switch over to biological response, sensitivity, and the adaptive capacity to ocean acidification. And we're going to be starting with Dr. Courtney Claypack, who is actually a postdoc at my institution, Mote Marine Laboratory. She completed her PhD at Old Dominion, and her dissertation research integrated coral biology, molecular ecology, and ecophysiology with environmental data and field experimentation, studying mostly two Pacific massive corals, but now she's down in the Florida Keys studying our Caribbean and Atlantic corals. And so she's going to give the first talk on biological. So Courtney?
Can everyone hear me? Great.
Yes, you're good.
Cool. All right, thanks. All right, so like Emily said, I am a postdoctoral researcher at Mote Marine Laboratory down in the Florida Keys. And so for this talk, I'm going to focus on the biological response of reef building corals under ocean acidification and focusing on their sensitivity and potential adaptive capacity. Next slide. So before I talk about the biological responses under OA, I just wanted to take everybody on a little journey and investigate further what is a coral. And so when you're looking at this photo on the right, you recognize it. It's a coral reef ecosystem full lush of life and corals. And a coral is actually a symbiotic organism where the coral animal has these tissues, and within the tissues of the coral animal reside single-celled photosynthetic algae. And this symbiosis is kind of structured around the nutrient exchange between the coral host and the algae that are kind of commonly referred to as zooxanthellae or zoox. And the symbiotic exchange is where the zoox kind of have a place to live within those coral host tissues, and in exchange provide photosynthetic byproducts in the form of sugars. And the corals actually take this, it's up to 97% of their daily metabolic intake, and they take this byproduct and actually use it to sequester calcium carbonate or calcify their skeletons. And so it's through this really obligate and beautifully successful relationship between the two that corals are able to grow and build the reef framework of coral reef ecosystems. And so this is happens worldwide, but specific to this meeting next, please, is Florida's coral reefs, formerly known as the Florida Reef Tract. They recently changed the name, so I'm going to be calling it Florida's coral reef. Florida's coral reef is the third largest barrier reef in the world. It extends from the central east coast of Florida all the way down just past the Florida Keys. And Florida's coral reef attracts over 16 million visitors per year. It supports over 71,000 local jobs, and it's valued at greater than $6 billion annually. And so in addition to these kind of economic services, they also provide ecosystem services that Janet touched on a little bit in the last talk. Most importantly, shoreline protection, especially down here in the Florida Keys, as well as critical nursery habitats for key fishery species, in addition to tourism, as well as pharmaceutical products. Next. However, in the past 4 decades, Florida's coral reef health has been in decline. And so that upper photo of Carey's Fort Reef that was taken in 1975 is your picturesque, characteristic, healthy coral reef ecosystem. But in the past few decades, the health of Florida's coral reef has been in decline. And so a lot of reefs, especially the same reef taken a few years ago, it is devoid of life and severely degraded. And so in this figure on the right, you can see that around the mid to late 1990s, mean stony coral or reef building coral cover has declined to about 5 or 7%, and it's stayed low. And it stayed really low as a result of a variety of stressors, such as ocean warming, ocean acidification, infectious disease outbreaks, pollution, overfishing, nutrient runoff. And so all of these stressors kind of in combination and separately have kind of kept Florida's coral reef from the ability to recover. Next, please. So for this conference and for this talk, I'm just going to be focusing on ocean acidification, or OA. And OA is proposed to reduce calcification rates of corals by reducing the saturation state of aragonite, which is that mineral form of calcium carbonate that corals use to sequester their skeleton. And with a rise in OA comes an increase in energetic cost of biomineralization or calcification, as well as an increased cost of acid-base regulation. So to kind of get an idea and understand where recent research has been trying to investigate and explain OA's effects on coral biological processes, both within the coral host and the symbiont, I want to take a closer look into the coral organism. And so if we look at this picture on the right, you will just take a look into one coral polyp. And so one coral colony is made up of thousands of polyps, kind of like the photo behind me. And if you look within the tissues of one coral polyp, you can see that there's this really complicated but sophisticated organization of like kind of compartmentalization of both the coral host, its cells, as well as the zoox or symbiont cells that reside within an organelle inside the coral host cells. And in looking at this compartmentalization, you can also see that the pH within the coral tissues is very different depending on where you're looking. And so recent research has actually found that corals are able to mitigate acidification of the surrounding seawater by maintaining an elevated pH and aragonite saturation in the subcalicoblastic medium. If you look at that photo, you can see kind of that red region. It's between the coral and the actual skeleton. So this is where calcification occurs. And corals are really good at keeping that pH and aragonite saturation high to basically promote calcification. In addition, the symbiont cells reside in that specialized organelle and host cells. And what the host or the coral host can do is they have a couple of carbon concentrating pathways where they will actually provide dissolved inorganic carbon to the symbiont organelle to promote photosynthesis. So if you look around that symbiont cell, you can see it's green. The pH of that organelle is actually 4, and it's to promote photosynthesis. And so by way of carbon concentrating mechanisms and photosynthesis, the symbionts are able to consume any excess CO2 that is entering the coral. And so as a result of this really intricate acid-base homeostasis, corals are able to cope with acidification levels that are more severe than what we're actually predicting for climate change, which is like levels of 7.4. However, it is very likely that this buffering capacity could eventually be exhausted, and we don't really know what the potential energetic trade-offs might be as a result of prolonged exposure. So this is where MOAT comes in and my research comes in. Next, please. I work within the Coral Health and Disease Program of MOAT down in Florida Keys. And down here, what we do is we conduct resiliency screening experiments. And so what we do is we are exposing many individuals of multiple species of corals over the past 5 years to 2-month exposures of a variety of treatments. And so these treatments are control, ocean warming, ocean acidification, and combined combined ocean warming and acidification at predicted end of century levels to understand all of our land and field-based nursery coral responses to these stressors, as well as determine, are there any individuals or species that are more resilient? And then we can use those corals in our restoration as well as our reproduction pipeline. So during these experiments and afterwards, we assess the physiology of the coral's response to these treatments, and we can do this during and after. And so what I'm about to show you is some recent results that we're working on publishing very soon. And so next, please. To orient you to the results, so basically we were exposing 2 coral species to those 4 treatments I mentioned. And so in this figure, to orient you, this is a principal component analysis, which is how those physiological traits that we measured during and after the experiment lay out into space with regards to treatment. So control is going to be those blue colors. OA is going to be red, ocean warming is yellow, and ocean warming and acidification is gold. And each point is a coral, and each point is laid out in space based on that physiological response of whatever we measured. So those black arrows, so we measured things such as Zoox density, their chlorophyll content, photosynthesis to respiration, coral host proteins, as well as coral growth. And so where these points lay out in space kind of tells us how these corals are responding to treatments. And so for both coral species, after 2 months of exposure, we found some really surprising results. And so we found that we found a negligible on the left and actually a positive effect of OA on our coral's physiology after 2 months of exposure. And this is because that red ellipses is kind of going in the direction of those arrows. However, when in combination with ocean warming, we found a negative effect. So that gold ellipses is actually shifted away from physiology, meaning that there was a negative response to OA and OW. So kind of baffled by why we got these results, we kind of investigated a little bit further. And what we realized was that our land-based corals that we used in these experiments were actually grown in water that had a naturally low pH anyways. So our OA conditions weren't really stressful for our corals. But this is kind of promising because what we've been inadvertently doing is raising our corals and hardening them and adapting them to more stressful conditions so that these are the corals that we have on land could potentially be resilient to changes in pH and mostly pH when they're outplanted to our restoration sites. In addition, I found a recent study that looked at these same coral species, but they were taken from the wild, and they did have decreased calcification under elevated temperature and PCO2, kind of similar to our OA-OW responses. Next, please. So how does this translate into what's going to happen with corals under future and rapid climate change? Well, first and foremost, it's probably very improbable to find a reef that is just experiencing ocean acidification. And just like Janet was saying, a lot of these stressors are acting in conjunction or in synchrony. And so it appears through our results and a lot of literature review that I've been conducting for this work is that corals are highly sensitive to both ocean warming as well as ocean acidification. And so reduced coral cover is typically driven by increased thermal bleaching events. And bleaching is where you have a breakdown in the symbiosis between the coral host and the zoox or algal symbiont. The symbiont leaves, the coral appears white, aka bleached, and they lose that nutrition. So essentially, eventually they could die if the stressor is prolonged. When the coral dies, in this figure you see here on the right, when the coral dies, it's basically open surface area for things to grow, such as microbial biofilm will form on top. And in conjunction with the indirect effects of ocean acidification, that metabolic activity is going to cause coral skeletal decay. And then we're going to result with a structural reef change where we're going to be losing that rugosity, that topographic complexity that we were talking about in coral reefs. And so that is going to change, and that is going to leave a reef more degraded. And what this study that was conducted, they found that even degraded reefs are more susceptible. So basically open, dead, decaying corals are going to erode much quicker and bio-erode much quicker. So I mentioned earlier that Florida's coral cover is only at 5%. And so of the corals that are remaining, if they're susceptible to ocean warming and ocean acidification, they're going to leave space for macroalgae and biofilm. And what we've actually been seeing in recent years is kind of a phase shift from coral dominated communities to now sponge, macroalgae, and soft coral communities. And all of those ecosystem services that coral reefs provide, such as shoreline protection, critical fisheries habitat, pharmaceutical products, tourism, all of that is going to be lost with the increasing degradation of coral reefs. Next. And this isn't just occurring within Florida's coral reef. This is actually happening at a global scale. And this recent meta-analysis that was just put out investigated the global coral reef net carbonate production under various carbon emission scenarios, both within the next 30 years as well as the end of the century. And again, they found that corals are sensitive to both ocean warming and acidification in conjunction, so much that they're unable to maintain this positive net carbonate production. So if you look in all of these panels, past, present, whether it is a lower emission scenario or a greater emission scenario, eventually net production is going to stop and actually decline. And they found that carbonate production actually will be decreased by 71% in 2050. And this also has potential cascading effects on other coral reef ecosystem processes, such as coral reproduction, as well as coral larval recruitment. If they're not growing, they're not investing, back to that energy trade-off, they're not investing their energy into reproducing. And there's other consequences of larval health under OA and ocean warming. And this study also found that by 2100, although 63% of reefs will continue accreting skeleton, most of those reefs will actually be eroding. Next, please. So I don't like to be a bearer of bad news. I'm usually a happy, bubbly person. So I'm going to leave my talk with some few final thoughts, research directives, both in the field and kind of where MOAT is coming into play. And so our research focused on 2-month exposures. And we know that ocean acidification is a process that's kind of happening year round. And ocean warming is restricted to those summer months. So we still kind of need to figure out what are some effective timescales and dosings to understand OA or the individual effects of OA on coral health. And then kind of found by our research and others is that some coral species are less affected by OA and OAOW. And so it's really important to keep kind of doing these resiliency screenings and monitoring to determine which coral species are less affected, because those are probably going to be the representatives of future coral reefs. And lastly, and this is relevant to my previous research or my expertise, is that all around the world and possibly within Florida's coral reef, there's going to be these reef sites that are going to be more variable. They're going to be more variable in their temperature. They're going to be more variable in their pH levels. And typically corals from those sites are going to be more resilient. And so it's really important for us to find reef sites containing these resilient corals. So reef sites that are naturally more stressful, because we can use those for 1, protection, and 2, potentially restoration efforts where we can outplant those and hopefully seed nearby degraded reefs. And that's also what is really cool and important about being with MOAT right now, is because all the research that we're doing is hopefully promoting and buffering this functional loss of ecosystem services in Florida's coral reefs by at least finding the corals that are going to survive or at least be resilient, and we can put those out and then also use them for selective breeding purposes. And with that, I will take any questions.
That was great, Courtney. Thank you so much. I'll start off with one question, while again, if you could kindly put your questions in the chat box, and then we'll be able to read them to Courtney and she can answer them that way. But I'll go ahead and start off. You did mention that at MOAT, we are also looking at disease as a factor. And obviously that's been in the news for the last couple of years with the skittle disease that's been going through. Do you have any thoughts or any preliminary data on how OA may affect disease or disease, against OA progression, since we are looking at that at MOAT as well as another variable.
True. Yes.
Yeah.
And I was gonna say, as like one of our collaborators, she's actually working on the disease portion of this. And I don't know, maybe you can jump in. I'm trying to remember. I think corals sometimes I think it's a very species dependent thing. That's what I've learned in all of this, is that some corals that are more susceptible to ocean warming and ocean acidification are equally as susceptible to disease, but then that doesn't always hold true for other coral species. And because in this study, the results that I showed, we actually took some of those pieces of corals after exposure and then expose them to disease. And I wish I could just pull up the paper right now, but I'm honestly not sure. I don't remember what her findings were. Do you?
Yeah, no, that's very accurate. It depends on the species. It also depends on the type of disease too. There was one study that was done where just looking at black band disease and the growth of it was actually slowed down by reduced pH, which was interesting and it didn't get examined any further than that yet. But yeah, that's it. Those are great points. And so again, please put your questions in the chat, and we do have a question here for you. What is the pH or PCO2 of MOAT's supply water? We hypothesized a similar acclimation since our lab coral water at its highest PCO2 was 650 and lowest pH at 7.6. She would love to chat sometime about it more.
Yeah, awesome. Of course. So yeah, it was kind of similar for us. So our exposure pH levels for OA was 7.7. However, our corals probably naturally fluctuate between 7.7 to like 7.8. So yeah, it was pretty much the same as the OA temperature or the OA treatment. And so that's why surprisingly we didn't find the potentially negative effect, especially on processes such as calcification.
Yeah. And I'll add a comment to that. Because that question came from Cheryl. We have changed the system a lot since the study that Courtney talked about, and we can give you a lot more detail, and things have changed. And our source water is at ambient, what is considered ambient conditions, but keeping in mind that the levels on the reef themselves change seasonally and daily and stuff as well. Yeah. Next question. What is your perspective on how grim the outlook for cold water corals might be?
Oh gosh, I can't answer that. I'm a tropical girl. I don't like cold. My bread and butter has always been tropical corals and typically under ocean warming. So I have to apologize that I'm pretty ignorant to the cold water world.
No worries. I'm in the same boat. Okay, so have you done any work with change salinity? Given climate change predictions for the Southeast, could a decrease in salinity directly affect corals?
I definitely so. I started with MOAT back in February. A lot of the work these results that I presented is work that was done before my time. So us in particular, we have not switched gears. We have not included a salinity component, although I would not be surprised if a change in salinity also is affecting what's going on with our corals. And so that's the beauty of these experiments, and having, we have an ocean acidification team as part of our coral health and disease. And so they're the ones that are championing, measuring water chemistry, looking at water dynamics. And I wouldn't be surprised if we saw decreased salinity having a potentially negative effect on corals as well.
Great, thank you. Next question is, what does adaptation of your land-based corals to lower pH suggest about timeframes for natural adaptation of corals?
Oh, see, that's where we need to continue doing research, right? And this is also contingent on the work that I've done in the past, is, there's a difference in the timescale. So corals, especially with growth is a very plastic trait in corals. And so it can adjust to the environment very quickly. And so even though our corals have been grown at naturally lower pH levels, what happens when they're put out at reefs that have potentially higher pH levels? Are they going to be more plastic in their growth and adjust and lose that hardenedness to low pH? We don't know. And that's actually kind of where my research within moat is kind of moving forward is we have these corals in our nurseries. We've been testing them to see how well they might perform against climate change stressors. But once we put our corals out, we just monitor them. We don't know what's changing after they're out in a variety of environments along Florida's coral reef. And so what I plan on doing next, and actually next year, is bringing some of those corals actually back into the lab to assess, is anything lost? What is the influence of their new environment as opposed to where they were raised? And so that's actually a really great question, because that's actually where our research is headed.
That's great. Thank you so much, Courtney. And I don't see any other questions for you. So thank you for that. And we'll go ahead and move on to our next speaker in the biological response section. So we're going to be now hearing from Ed Sherwood, who is the executive director at the Tampa Bay Estuary Program. He became Tampa Bay Estuary Program's executive director in 2018 after serving as the program scientist since 2008. He is responsible for maintaining Tampa Bay Estuary Program's strong interlocal partnership and continuing the Bay's science-based restoration and recovery strategies. And he's going to talk a little bit about the seagrass efforts and some of the acidification stuff that's going on there. So thank you, Ed, for joining us.
Morning. Can you guys hear me okay?
Yes, great.
Yes, I appreciate the opportunity to speak to you all this morning. As a National Estuary Program, the focus of this SOCAN workshop of communicating and increasing our awareness on ocean acidification issues is aligned well with our comprehensive conservation management plan for the Tampa Bay Estuary Program. And really that CCMP is what drives much of the work that we do in our region to get the Bay back on track in terms of increasing its resiliency to future perturbations such as ocean acidification. So you can go on to the next slide. So yeah, I'm gonna be talking mainly about our core initiatives that are focused on increasing our understanding of ocean acidification within the estuary and what we can do about it and help mitigate the impacts of that of observed ocean acidification along our coastal environments and in the estuary in the future. So you can go ahead and go to the next slide. A lot of what you heard earlier from Dr. Reamer and Dr. Klapek, I'll kind of summarize in these couple of slides, but the complicated thing in estuaries as it relates to ocean acidification is the effects of nutrient pollution, as well as sediment conditions, as well as riverine discharges into these nearshore water bodies. So there was a great overview of why the world's oceans are acidifying because of the increase of CO2 in our atmosphere and how that's diffusing into the coastal oceans. But really the stressors along the coast can exacerbate acidification issues within estuaries, and that creates a lot more variability in the estuarine environment, and that could cause additional stressors for a lot of the biological communities that we think are important to maintain along our coast. So you go ahead to the next slide. I'll just highlight on this slide, this basically summarized a lot of what Dr. Reamer and Dr. Klapek just mentioned. There's going to be winners and losers as it pertains to ocean acidification. On this slide, I'm basically highlighting losers. Those organisms that might be facing more stressful environment to maintain their life history strategies in the future with an acidifying coast. In particular, in our region, a lot of the crustacean larvae, particularly stone crab and blue crabs, are of particular interest and I think research interest from our standpoint. And we've been working with Moat Marine Lab as well as researchers from Florida Southern University, Dr. Phil Grevenese, to understand the implications to stone crab larvae and whether or not some of our habitats might be offering refugia to these larvae in the future. That sort of strategy of enhancing coastal resilience with seagrasses and other blue carbon habitats, and I'll touch upon in a little bit, is sort of core to how we're trying to look at combating ocean acidification issues in the Tampa Bay Estuary. And as Dr. Claypick just mentioned, the increasing stressful environment of increasing temperatures and lower pHs are causing all sorts of issues for some of the reef tracts in the Florida system. We're actually starting to see some hard corals infiltrate into the Tampa Bay region. So that has, I think, sort of long-term management questions in terms of maintaining these important coastal ecosystems in the future, of understanding if certain areas can offer refugia to ocean acidification impacts in the future. So if you go on to the next slide, I'll kind of highlight what Dr. Reamer was touching on earlier about the Tampa Bay Estuary. We've been trying to maintain a certain ecosystem balance in the estuary over what historically has happened in the Tampa Bay system. So we know back in the early 1970s to 80s that we had really bad eutrophication issues. We had a lot of nutrients going into the bay that was causing a lot of algal blooms, and we had a significant loss of seagrasses. So the system was very dystrophic. It was out of balance at that point. We did have higher pHs because of that higher primary production, but the swings were much more significant than what we're seeing now. And through concerted effort of managing nutrient loads to the bay, we've seen increasing seagrass resources, and that has offered a coincident increase in pH through time, as shown on the graph here. And really, as we move towards managing the system in the future, is can we continue to create some resilience to these increasing stressors along our coast by maintaining key habitats in the Tampa Bay Estuary, namely seagrass resources. And this is being investigated in a number of other areas along the West Coast and in the Northeast. It's, increasing kelp forests and eelgrass beds for the benefit of combating coastal ocean acidification issues. And Dr. Reamer highlighted, the benefits of what plants do in terms of utilizing the CO2 in the water to create more resilience to lower pH conditions within estuaries. And some of the research we're focused on now in partnership with Dr. Kim Yates at USGS is really understanding the benefits of restoration of these key habitats in our estuary. There's significant differences between bare sand habitats and seagrass habitats in our estuary in terms of the buffering capacity of pH within the estuaries. And Dr. Gravinis' works is looking at larval recruitment into seagrasses versus bare sand habitats, and whether or not that creates some resiliency to larval recruitment success. So again, that's one of the things that we're trying to continue to encourage from the standpoint of managing the system as a whole. We have seen some recent declines in seagrass resources along the Southwest Florida coastal estuarine systems, all the way from Tampa Bay down to Charlotte Harbor. And that, is a concerning thing for us, because that could potentially be reducing our buffering capacity to future ocean acidification issues. And certainly the importance of these habitats are paramount for our system to become more resilient and offer refugia to some of the important biota that might be impacted by acidifying conditions along the coast, Gulf Coast in the future. We go on the next slide. Another core strategy that we're focused on is maintaining blue carbon habitats. Was mentioned a little bit before. We're fortunate in the Tampa Bay region to have 3 of the blue carbon habitats that offer additional climate mitigation benefits in our region. These are habitats that offer huge carbon sinks for atmosphere, increasing carbon CO2 concentrations in the atmosphere. These habitats, will absorb those CO2 and then ultimately bury those in sediments. And while the carbon circulating in the ocean is a fairly significant amount of our CO2 global CO2 budget, these habitats only represent about 2% of the land area along the globe, but they offer disproportionate amount of carbon storage in the future. So again, this strategy to maintain these habitats in the future is going to be paramount for our maintaining resilience in the Tampa Bay Estuary, as well as in the global environment in terms of buffering future climate change impacts, as well as ocean acidification impacts. So we have done some preliminary research and understand that we might be seeing a tipping point of these habitats around the 2070 timeframe, where we might start seeing a loss of some of the emergent tidal wetlands, namely mangrove and marsh habitats, with increasing sea level rise and a continuing coastal area that's urbanizing and expanding in terms of population. And the only saving grace would be potential increases in seagrasses at that point, but that's going to take a very concerted effort to maintain water quality conditions in a deeper estuary at that point with higher sea level rise rates in order to maintain those seagrass blue carbon habitats in the future. So not to be pessimistic in terms of outlooks, but it just heightens the need to continue to maintain these important blue carbon habitats in the future for our future well-being, not only within the Tampa Bay Estuary, but globally. Go ahead and go on to the next slide. So as I mentioned, our core strategy has just been to raise awareness of what pH conditions exist in the estuary relative to the near coastal environment. So we've been working with Dr. Kim Yates at the USGS to establish some long-term monitoring stations within the estuary itself. So those black data points represent a site that was established back in late 2017 and has continued to be maintained in the lower Tampa Bay region. And then we also had some resources from the US EPA and the Tampa Bay Environmental Restoration Fund to have a complementary site offshore, and that's signified by the red data points in the graph in the middle. You can see, there's a lot more variability in the pH conditions within the estuary relative to the near coastal environment. And what long-term trends are in terms of the near coastal pH conditions and, our changing estuarine environment might mean for those swings in pH in the future is why we're establishing these sites, concomitant with, the changes in seagrasses, mangrove, salt marsh habitats. The buffering capacity of our estuary might become reduced in the future, and it's important to capture that through these long-term monitoring instruments. So if you go into the next slide, it also highlights sort of the benefits of real-time monitoring. That site that was established in the Tampa Bay Estuary, the lower part of the Tampa Bay Estuary, was very close to an area called Piney Point. Many of you might have heard of what happened in terms of an emergency discharge that occurred from Piney Point. This basically gave sort of a test case of what happens when you load an estuary with a lot of nutrients. It stimulated primary production of key phytoplankton groups right at the start of this discharge, which is highlighted on the graph on that yellow bar there on the center right. So we saw immediate diatom bloom that occurred in response to that nutrient load. And we saw changes in both CO2 and DO conditions within the estuary, as well as pH conditions in response to that large slug of nutrients. So this just heightens and really exemplifies the complexity of what happens in the estuarine environment in the face of nutrient loads. So these punctuate events that in this case were caused by a human anthropogenic impact stemming from an old mining facility. But there's also loadings that occur that Dr. Reamer was highlighting in response to hurricane and storm events that are loading the system and really driving some of the pH conditions within the estuary. You'll notice later on in the summer and towards the end of July, that's when we had a real explosion of red tide bloom within the Tampa Bay Estuary that we were also suspecting that was responding to the latent nutrient load that was within the system stemming from that event. And you see this real high primary production signal in both the pH conditions as well and DO, but then also this crash after that bloom subsided and started being consumed. So again, this highlights the complexity of what's happening within the estuary and how nutrients might be impacting our ocean, our acidification conditions in the future. So if you go on to the next slide, just want to summarize, all these are important for our management of the system in general, and our continued focus of restoring seagrass habitats and important mangrove marsh habitats not only has these benefits for creating resilience in the system and buffering potential acidification issues, but it also has economic benefits to the region in terms of a clean and healthy bay. We're driven by a tourism-based economy, and if we're starting to see a decreased resilience of the system, similar to what's happening in the Inner River Lagoon, in terms of these habitats, that makes ocean acidification issues more of a vulnerability for some of the key biota that we've come to rely on in terms of our economy within the region. We're now starting to see commercial shellfish operations starting within the Tampa Bay Estuary, and a lot of that, the success of those operations are contingent on maintaining healthy ecosystems that could support those in the future. Those are namely the seagrass beds and those mangrove marsh systems that are creating that resilient Tampa Bay ecosystem. So if we continue to lose those key habitats, the dystrophic conditions I mentioned in the beginning of my talk might become more prevalent and cause it to be more of an issue in terms of allowing those commercial operations to persist. So again, our hopes is to raise awareness among our citizenry and our management conference to continue efforts to reduce nutrient loads to the bay for the benefit of these key habitats, while also restoring those other blue carbon habitats so that we can continue to mitigate these potential climate change impacts stemming from ocean acidification, as well as other issues that we face through our resource management of the estuary. So I'll stop there and welcome any questions you might have.
Thanks, Ed. That was a great talk and really introduces the complexity of these issues in our estuarine and coastal ecosystems. Again, if you guys have questions, please type them into the chat so I can read them to Ed so he can answer them for you. But I'll go ahead and start with a question. You mentioned in the beginning talking about areas of refugia, which I know in the ocean acidification world seemed to be pretty important. We look for a lot of areas of refugia for our corals, but looking at these estuaries as refugia, not only for the carbonate chemistry itself, but for different organisms like stone crabs or our clams and scallops and stuff, at least in the state of Florida. You mentioned that a lot of the stuff with the seagrass recovery was really focused around reducing nutrient inputs. Were there also efforts to actually put in new seagrass beds, the old, kind of the more traditional way of doing restoration efforts? Did that happen at all in Tampa Bay?
There was a little bit, but primarily the recovery of the seagrass resources were a result of cleaner waters through less nutrient loads into the Tampa Bay ecosystem. There are areas out west that are looking at kelp restoration, direct kelp restoration to combat ocean acidification, also along the northeast. But our success has been largely driven by controlling nutrient loads to the nearshore environment. Also, I probably failed to mention sort of the uniqueness of our Southwest Florida coast is the spring-fed rivers that enter our estuary. And I think some recent work by USF's Sean Beckwith kind of highlighted that we might have calcification sweet spots for certain organisms because of those spring-fed rivers that are contributing calcium carbonate to the nearshore environment. That, in combination with the stable and persistent seagrass beds along our spring coast and our Southwest Florida estuaries, again, might prove to be a novel refugia for certain organisms to really create more resilient environment in the future in the face of continuing global ocean acidification. But if we lose those habitats, the buffering capacity of the rivers might be diminished. So all that has to be understood and monitored in the future. And I think that's where a lot of our research is focused, particularly in some of the work we're interested in terms of understanding how resilient the Tampa Bay system is now with the restored seagrasses we've experienced over the last 3 decades.
Great. Another question has popped up. Do you have any information yet on the impacts of oysters and other filter feeders in Tampa Bay?
No, not as it stands right now. That's another research gap that we hope to focus on. As I mentioned, we have some commercial fisheries, and we have now a commercial oyster operator in the lower part of the bay. So we're hoping to expand upon our partnerships with some of the growers to understand what it might mean for them in terms of their business model and what ocean acidification might pose, what risk might ocean acidification pose to them in our region. I know, especially along the Pacific Northwest, they have much different acidification issues than we have here, and it's already impacting particularly oyster shellfish growers in the Pacific Northwest. We're not sure that's happening for some of the operators along the Gulf Coast and Southeast US, but that's definitely a research gap that needs to be addressed.
All right, thank you. And I don't see any other questions at this time popping up, so we'll go ahead and say thank you, Ed. And that does conclude our biological response section. And we're going to go ahead and move on to our social vulnerability, impacts on communities and their adaptive capacity. So our first speaker in that group is Jennifer Hecker. She's the executive director of the Coastal and Heartland National Estuary Partnership, which used to be known as the Charlotte Harbor National Estuary Program. She's responsible for working with diverse stakeholders and local and state and federal government to restore and protect the water resources in the CHNEP area in Central and Southwest Florida. And so with that, we'll go ahead and pass it over to Jennifer Hecker. You're muted, Jen.
I just unmuted, but I got muted again. So hopefully I'll stay unmuted. Thank you guys for having us. We are, like Ed, part of the US EPA National Estuary Program. Next slide, please. And our National Estuary Program service area encompasses 5,416 square miles and includes 7 different estuaries along Southwest Florida, going from Lemon and Donna and Roberts Bays in Sarasota County down to Estero Bay in Lee County. It also encompasses inland and coastal communities, including 10 counties and 25 cities, and you can see them outlined here in the aqua. And we basically go up and wrap around the eastern side of the Sarasota Bay Estuary Program and the Tampa Bay Estuary Program, because we go to the headwaters of the Peace River, which flows down into Charlotte Harbor. Like Ed mentioned, we also are guided by our comprehensive conservation and management plan. And in the CHNEP collective CCMP plan, we have 4 priority action areas of water quality improvement, hydrological restoration, fish, wildlife, and habitat protection, and public engagement. And of course, climate change affects many of those priority actions. And with regards to public engagement, that's what we're really going to talk about in this quick talk about the intersection between biological science and social science. Next slide, please. So we did a climate change vulnerability assessment, and this was required by the US EPA of all national estuary programs under their Climate Ready Estuaries initiative. And what we did is we looked at what climate risks were anticipated for the Southwest Florida region and how those risks might impact our objectives in our CCMP plan. And the way we went about it was we surveyed 5 experts and approximately 50 plus public participants in a public workshop. And the public participants first created a list of climate risks, and then they ranked those collectively. And then we sent those to the experts who ranked those independently. And they looked at both the likelihood of that risk occurring, and if it did occur, how severe of an impact it would create on the objective. So you can see here to the right, the list of the experts from our area that we interviewed for this purpose. Next slide, please. So the purpose for us to undertake this was to identify those climate change risk factors that may inhibit us achieving our CHNEP, CCMP objectives, and to identify disconnects between the public and the climate experts to understand the knowledge and understanding of these climate risks among the public, and to identify where there might be possible research needs as well, where there was no scientific consensus amongst our experts. So here to the right shows a graphic from one of our risk factor assessments. And you can see here the grouping of the symbols in the upper right-hand corner, which illustrate the different experts and where they rank the likelihood of occurrence and the level of impact to the objective. And also then the P, which represents the public. And here you can see there was a disconnect where they both agreed there would be a high level of impact if this risk, did occur. But on the other hand, there was a big disconnect between the experts and the public because the experts thought it was very likely and the public thought it was very unlikely. Next slide, please. So diving into specifically those related to ocean acidification, we just had a couple of them. The first one was looking at fish and wildlife protection and our objective to protect, enhance, and restore native habitats where physically feasible and within natural variability. And we looked at, well, what was the risk? Ocean acidification may impact shellfish and the habitat they create. And how would that, if that occurred, impact us achieving that objective to protect fish and wildlife and restore these native habitats? So the experts ranked that ocean acidification may impact shellfish and the habitat they create, and they ranked it as medium to high impact and medium to high likelihood of occurrence. The public ranked that this would have an impact, but again, in this example, they ranked it as a low likelihood of occurrence. Next slide, please. The other ocean acidification related assessment we did was looking at ocean acidification impacting shellfish populations with regards to water quality improvement, and our objective that by 2025 that we meet shellfish harvesting standards year round for the Myakka River conditionally restricted area and the conditionally approved areas of Lemon Bay, Gasparilla Sound, Myakka River, and Pine Island Sound. And you can see here there was mostly agreement upon the experts to rank medium to high level of occurrence and level of impact, and the public who also picked medium level of occurrence and high impact to the objective. So that was an area where you saw generally more agreement in the water quality versus the habitat. Next slide, please. So really the takeaway is, and this made sense to us based on our interactions with the public in our region, is that ocean acidification impacts on water quality are generally better understood by the public than the impacts on aquatic habitats. And so we, really looked at that as an educational need, that we really need to do more to explain about ocean acidification and sea level rise impacting habitat, particularly the loss of existing shallow water and coastal habitats, including shellfish habitats, and raise the awareness that this is a risk that is likely to occur in Southwest Florida. So there's a lot of public communication and outreach and education that's needed on that front. And we also identified that more research on ocean acidification in Southwest Florida needs to occur to help raise that public awareness and understanding. Next slide, please. So just in concluding, I want to say that we actually have an event coming up in April, the Southwest Florida Climate Summit. It's our second annual event, April 7th and 8th, and we're inviting all of you to join us. It's going to be a hybrid event at the Collaboratory in downtown Fort Myers, as well as a virtual event. And registration is required and will be available. It's free. It'll be available on our website in February at chnep.org. Next slide, please. And then if you want to learn more about our comprehensive conservation and management plan, you can go to our website, chnep.org/our-plan, or to read the full climate change vulnerability assessment. And there's a number of other, I think there was up to 30 or 40 different risk factors that were assessed. If you'd like to look at that further, that's available at chnep.org/publications. I know that was quick, but hopefully that covered the general things that you all were looking for, and happy to answer any questions you have. Thank you.
Thank you, Jennifer. I know that the social science aspect can be very challenging to explain, and I think you did a really great job. So again, thank you. We'll take a couple of questions from you before we move on to our next speaker. I guess my question would be, how did you reach your public? How did you get responses?
So we actually organized a public workshop. It was a day-long event to go through climate change science and then try to talk about with the public their understanding. And again, that we did find that there was not a lot of science specifically on acidification at that time. This was now a couple of years ago. But nonetheless, just working with the public participants to try to identify what they saw as risks related to climate change based on their fundamental understanding of it as it pertains to our region. And I saw there was a question in chat about, the immediacy. And I think that is definitely part of the issue, is that until people experience some of these impacts, they're not really relevant to their daily lives. But we found that particularly difficult with things like acidification that are a little bit further afield from what people are experiencing than, for instance, the increased temperatures in Southwest Florida, which they're already experiencing, the flooding from increased storminess and sea level rise. So those are typically the climate change risks that people better know about and understand. Acidification is something that we feel there's a lot more work to be done.
So do we. Next question is, did you look into what was causing the difference in ratings among experts? And did that help identify any research priorities?
Yes, it absolutely was helpful. This whole exercise was really interesting. We, went in with just looking at how it was going to impact our CCMP, but really those educational and research findings were extremely helpful in guiding our updating of our CCMP. And where there was lack of consensus amongst the experts, we definitely felt that indicated that there was more research needed specifically to our region. I think that was part of the challenge, is that sometimes we had experts that were drawing upon different studies or experiences. And this was talked about at the beginning of the day, is that really we can't always extrapolate what is happening in one system with another. And so we felt that could have attributed to the differences in their responses and the fact that we didn't have specific regional data to help guide them.
Excellent. Thank you very much. I think we're going to move on. Emily, who's our next speaker?
Yeah, thank you. Our next speaker is Juliana Mullen. She is the communications manager at Naracoos and the community manager of the Ocean Acidification Information Exchange, also known as the OAIE, which I'm sure she will press many of you to go ahead and join. It is a great resource. She has spent much of the past decade at the intersection of science and communication, and she's worked on projects ranging from the development of a sustainability standard for benchmarking fisheries to creating lesson plans for 8th graders studying marine genomics. She started working on as the community manager for the OAIE in 2018 and has a lot of great information for us. So I will go ahead and pass it on to Juliana.
Hey, it's great to be here. Thanks for having me. Hoping you all can see me. We do next slide, please.
You're good.
Okay. So I'm Juliana. I'm the community manager for the Ocean Acidification Information Exchange, which is an online community for professionals who are interested in or working with ocean or coastal acidification. And initially, the hope today was actually that we would have Quinquet Marquetta Goodwine of the Gullah Geechee Nation present her work for you. And so I'm going to do my best to talk about the products that she developed through the OA Information Exchange. And she created a video that I'll show briefly, but next slide, please. I'll just walk you through a little bit of the background of this. So the OAIE is an online community. Our mission is to catalyze response to ocean and coastal acidification through the power of collaboration. And in 2020, we opened up a micro grant opportunity where any member could apply for up to $5,000 to launch a project that would advance the OAIE's community mission and also facilitate their own work related to OCA. And one of many shameless plugs, there will be another micro grant round, so I encourage you to join the site, if only for that. So Quinquet, like I said, was selected for funding, and she was going to create outreach materials, a video, infographic, and a lesson plan for K through 12, specifically designed to educate vulnerable communities about what coastal acidification was doing to the resources in the Southeast. And next slide, please. So hopefully if we click this video, you will be able to see what she came up with. Right now, we don't have any sound.
So the sound on the video is pretty quiet. I'm gonna go ahead and try again, but I would encourage just to increase the volume as well.
You have to make sure that the audio is actually shared when you're doing a screen share as well on Zoom.
Okay, I'm gonna go ahead and try again. You guys tell me if you're able to hear.
Now we can hear it.
This year to the Gullah Geechee Nation. In the sun, rise, we say, God, don't smile upon me. Yeah, this your way. We river and they go right on yonder to the ocean. Oh, welcome to Chile. Want you to take a little journey down this creek with me. We gather, we swim, and things like that. And why this year, concerned we, Amnesia Island, where we cast, we net, like we ancestors been to do.
I don't know why your audio went out again.
This young man, as you would say, what does this mean to all of us that enjoy this atmosphere, most of all, the ocean? Well, the ocean doesn't stay in one place throughout the day. And as the temperatures change with the climate and the things that we do on the earth continue to change and alter and go into the ocean, we now have a new word that, just like you might have never heard the Gullah language when I was speaking it, we never heard the word ocean acidification until they came from the scientists to our shorelines here in the Gullah Geechee Nation. Burning fossil fuels releases CO2 into the atmosphere. Bodies of water, like the ocean, absorb the CO2. This causes the ocean to warm and acidify, which is harmful to the aquatic creatures, or as we say, the creatures with DNAC. So now, the water's acidity levels can be affected by land pollution, by runoff from hard surfaces like parking lots and roads that we drive on, or changes in water particle composition that come from people putting out what I call destruction. And these things keep going into the sea, we won't have what we consider the delicacy, like oil crab, or we devil crab, or plenty of the visitors, like, but not just come here for sea, but nyampa, like a week. And this concerns me because the shells and skeletons of our shellfish, the oysters and the crabs slowly dissolves as the ocean acidifies. And their growth slowed. And so, if this concerns you too, I invite you to come join me on these shorelines, sea islands, and let's work together against climate change and ocean acidification. And let's make not only the sea and the ocean, but all of we healthy.
Great, thanks for sharing. So as you can see, it was a really great video. This is the infographic that was partially shown in the video, and that was also one of the project outputs. And I'm glad that Quinquet is here. I didn't realize that she could join us. So hopefully, if there are questions for her, she's obviously better suited to answer them than I am. Let's see. Sorry, I just lost my place. Let's go to the next slide. The OAIE microgrants also funded another project that was related to stakeholder outreach. And that was a video done by the Chugash Regional Resources Commission, specifically around sampling in South Central Alaska. And that video, the materials that Quinquet produced have all been shared on the OAIE. They are also available on YouTube. I can send the links afterwards if you're interested in them. But this is now, a place where we're building a library of resources for anyone who needs them, whether it's for stakeholder engagement or research or whatever the community is looking for. Next slide, please. I think a lot about the impact an online community can have in the real world. It's kind of my job to think about that. And obviously funding is a great way to get people able to be involved and able to collaborate and afford to engage. But I also wonder, a lot of the time, what kind of impact can be had when there isn't funding involved. And trying to quantify this, I gave a survey to members of the exchange, and I tried to kind of capture this value. How has interaction with the exchange catalyzed any real world collaborations? And actually 36% of respondents said that yes, their engagement with the OAIE had catalyzed something in, quote unquote, real life. And these were just some of the write-in answers that people gave. And it's really encouraging to see that this work, both the platform itself, our ability to fund microgrants so that people like QueenQuet can produce these great materials, is having an impact beyond just being a little bubble online. So I do think that these kinds of communities, and the OAIE in particular, can translate to real world impacts. Next slide, please. So this is my final shameless plug. There are 1,500 members. We're still growing. It's a community with all sorts of different stakeholders. We have regional teams, including the Southeast team. QueenQuet was working with people in the Southeast team and the content creators and educators team. There are teams based on regions and interests. So I really encourage you to come over, check it out, join. You don't have to be an expert. You can join whatever interests you, and you can help others by sharing your expertise, ask questions. You can get access to all of the resources I've talked about and others. And I think it's a great way to jumpstart or continue your work with stakeholder engagement. So thank you.
Thanks very much, Juliana. We're really excited to be able to share that video. SOCAN has been a big supporter. Well, we hope that we're a big supporter of QueenQuet and the Gullah Geechee, and we are active participants in the Ocean Acidification Information Exchange. So yes, we're going to join Juliana with our shameless plug of the OAIE. If you haven't joined, please do so, and make sure that you get onto the Southeast team. It's a great way to share resources, information, ideas, and collaborations. We already have a question popping up, and I will just add that please feel free to ask questions to Juliana, QueenQuet, and if we didn't get your question for Jennifer, we can do that now as well. So first question is, for excellent video and infographic, what type of responses or questions have you gotten from others who had the opportunity to learn from the video? And a big, we need more of this, double exclamation point.
Yes, I saw that. I'll take that one. This is QueenQuet, Gladdy Fadea with all 100 children. I have gotten excellent responses from teachers in particular, because we did share this not only on the information exchange, but we also shared it with the South Carolina Marine Educators Association, which I'm a part of. So a lot of the teachers were very appreciative because they didn't have any materials here in the Southeast to specifically address this. And they liked the interactivity of it so that people could actually go to the beach, go to the ocean and do things. And so I've heard from a lot of people, especially during the pandemic when people were to stay at home, if they lived in the Southeast, this was something they could also use and go out with their children and so on. So it's been really good to know that the video is of value to many people. And I think my greatest experience with responses was actually on social media, when I saw people who are oceans folks on social media saying, hey, I just found this great video from QueenQuet on ocean acidification. And their voices had the exclamation points about, it seemed, when they tweeted it and things like that. So it's always cool to not expect folks to really look at the work and then find out they're really looking at it, they're using it and everything. So I'm happy to know that this made a difference. And I have made some other things related to climate action as well since I did this. So about heat island effect versus the sea islands, so that people would really grasp that as well. So this kind of stepped me off into that and hope to do some more things like it. Glad y'all enjoyed it.
Thank you. Next question, I think this one is for Juliana. Do you post acidification activities for educators that would be easy to be used in the classroom on the OAIE site?
There's definitely been resources that are already available in the teachers, educators, and content creators team. I'm thinking of a couple things in particular that are already there. But one of the great things is if you're looking for something in particular, you can definitely ask if someone has something that they would recommend. There are, I'm not going to look right now, but I'm pretty sure that there have been lesson plans and other documents uploaded that would hopefully be of use. So short answer, yes.
I have another question for you, Juliana. Do you feel, think, or know if the OAIE has had any impact on either local, state, or federal government or management plans?
The OA Alliance is a group that's working in New York, and they have been very active on the info exchange. And I know that one of the answers I received, I think, was in reference to that particular task force that was formed and how the exchange had helped inform some of their work. So that was really exciting to see. Because we are a neutral platform, there's no lobbying on there or anything. I can't say for sure that it has resulted in a change of any policy in a one-to-one kind of way, but I do feel confident that it has helped inform people, educate, and there are a lot of different policymakers on the site. So hopefully it's a useful resource and can also help with some impacts in that regard, not just in the areas that I discussed earlier.
Great. Well, I don't see.
Can I add to that answer? Also, just because I work on a lot of United Nations things, I've also shared this with people at that level. So people are aware of it, not just within the United States, but also some other global folks who are in positions to do things on a global level as well.
Excellent.
Awesome.
We do have one more question for Queen Quet. What is the most difficult thing for educators to understand about acidification? That's a really great question, because I think that also tells us what it is that we're not communicating when we do our outreach.
Well, I can say it's not just educators, it's just everybody, right? Because people aren't accustomed to hearing that water is acidifying. So to clarify, what do you mean by that? Usually is the biggest question, because people hear it and they say, well, oh, the ocean's turning into acid. So people need to really understand that and understand ways to grasp the scientific information. So when we're speaking in terminology like CO2 emissions and pH, that's not in the average American vocabulary, the average person's discussions over the dinner table either, much less in the classroom. So having to go ahead and what I included was a glossary of different things as well, so that educators could have ways that these types of scientific information can be broken down into a more common vernacular, one might say, for them to use, especially with any grade level at all. And I think that that is what makes the difference with community members. If we can just not do techno speak, and I know that's hard for us as scientists, I'm a computer scientist and mathematician, I get it. But if we don't do that, the better off we'll all be, because if we can make this just a clear understanding of what it is that's happening in a way that a child could even understand it, the children will lead and they'll teach their parents and their grandparents. So the teachers' jobs will be that much easier with the topic. So I think that the greatest thing is that we have to focus more on ensuring that we clearly define the scientific terminology and then put it in quote unquote basic English when we present it. That's the thing that I think is going to be critical in terms of educating everybody.
Definitely. I know that depending on who I'm speaking to, Alka-Seltzer is not very popular anymore. Most people
Right.
are using Tums, but Alka-Seltzer, drop that little tab in water.
Yes.
That's a great way to show people, because that is a calcium carbonate or magnesium carbonate mineral, and you can drop it in the water, and the water causes it to dissolve. So maybe
that's a nice example.
Yeah, other people can go ahead and do that. So we're just about getting to the end of our morning session. I know my watch has been telling me to stand up and take a break. We really hope that everyone will come back after lunch and join the small breakout sessions, because this is where we really get the chance to have deeper discussions and understand where we're all coming from different perspectives. And I just want to say, we did not leave out the aquaculture industry on purpose. We just couldn't seem to get together to find someone to give a talk. But I want to point out that a SOCAN member and one of the members of the meeting planning committee, Victoria Parks, is helping moderate this afternoon, and she's from Sea Venture Clam Co. So please pick her brain and also challenge her to tell you from her perspective what's going on in the aquaculture industry. So basically at 1 o'clock, you just come back to the meeting, and Laura is going to separate us out into breakout rooms, and we'll give a little bit of introduction about the breakout topics when we come back for lunch. Also, we emailed you some of the breakout questions that we hope to address, so you are prepared with as much information coming into this afternoon as possible.